You spend a lot of time as a scientist waiting for incubations to finish. I have to be here for another 18 minutes because I'm waiting for these guys to finish blocking so I can do primary. So I figure out with that time, since I finished everything else I had to do today, I'll tell you the story of how I wasted a full month with failed experiments because I didn't understand the chemistry, the full chemistry behind an experiment that I was doing. I'm a cancer biologist, and I'm studying something called the alternative lengthening of telomeres pathway, which accounts for 10% of all cancers. And one of the main assays I use to test for this cancer activity is the C-circle assay. So I'm testing for this structure here. It's not directly this cancer activity, but it is a way of measuring the cancer activity. In the same way that you could, smell smoke and know that a fire is happening without seeing the fire, you can test for this and know that all is happening. So there's a 4-day assay that I do where I convert the C-circle into something called a G-strand, and then I measure light that gets output. I put it at the end. It's a lot more complicated than that, but I've condensed it just to explain this particular problem that I encountered. This is a really tough assay, and there are so many ways for it to go wrong. One of the ways for it to go wrong is to have the wrong pH. Now, this assay needs a pH of 7.6, pH being how acidic a solution is. And I get the pH to 7.6 with a buffer called Tris, which is right there. And a very time-intensive way of preparing a Tris buffer is by using the Henderson-Hasselbalch equation. You can use Tris base and hydrochloric acid. You got to do a bunch of math, and it takes a while to do. But you don't have time. There are so many experiments going on at once, so many things to do. It's very important to do things as quickly as possible. So what you use is something called Trisma, Tris base, and Tris HCl. I'm pointing at the wrong things. This is my first time doing this. And these are tables. The math is already done for you. You just have to measure out the weights, and then you have the proper pH once you add those powders to water. The trouble with one of the small troubles with that is that the powders themselves will be contaminated by water. You've got water in the air, humidity, which will go into those powders and make them slightly heavier. So if you you're trying to weigh out a particular number of grams here, part of that will be water, so you'll get the wrong weight. So it's important if you're not desiccating, meaning if you're not drying, you can use something to pull the water out of the powder. I don't happen to do that. If you're not desiccating, you need to use a pH meter to figure out the right pH. So the way a pH meter works is by measuring the difference in potential across two electrodes. So this is a single junction pH meter here. So this is the pH meter electrode, and then there's a reference electrode. So you actually put this in the solution, and then you can measure the potential across this by ions from the solution going through something called a junction into this cavity with this electrode, and you measure that potential there. And it requires this junction to be clear, to be open. So gosh, this is like upsetting that this happened. A single junction pH meter is very cheap. You can get it for like $50. A double junction pH meter is going to be $400 or so. So what had happened is our double junction pH meter broke, and we switched to a single junction pH meter. And this was when my experiments started to fail, and I didn't make the correlation because we made the switch, and then it had been like a month or two where I didn't have to make the buffer, and then I needed to make the buffer, and I made it, and after all that time, I figured that it was the buffer. I thought it was so many other things first. I remade all of my solutions, I remade all of my buffers, I repurified all of my genomic DNA, I changed so many things because I didn't think the pH meter was an issue, because everyone else in our lab didn't have problems with the pH meter. Everyone was using this single junction pH meter and not having problems. So I figure, why would I have problems with it if it's working fine for everyone else? Well, the reason I was having problems with it is because of an interesting bit of chemistry. So this reference electrode here is silver chloride, and this is specifically for amines, so ammonia. Silver chloride will react with ammonia to form this soluble complex here, this very small complex. Now my buffer, tris buffer, has this amine right here, and the same kind of reaction will occur with silver chloride, where we make this complex here, but instead of this small complex, we've got two of these on either side of this silver. So it's huge. This thing is huge. It falls out of solution, and it clogs this junction here, which prevents the potential from being measured across these electrodes, which makes the pH reading totally wrong. And the really important thing about a double junction pH meter is that we have this junction here, and then we have a secondary junction here. So this problem that I was having can occur with a double junction pH meter. So that's why it's so important to have a double junction pH meter. And I've actually recreated the problem for you right here. So the pH of this solution is supposed to be 7.6. And with my single junction pH meter, it's reading pH 9.66, which is really high. So if we go over here to my double junction pH meter, this is the new one that we got, we're reading pH 7.71. Can you see that? Yeah, pH 7.71. So that's a pH of 1.6 or so different across those two things. And that will ruin the reaction. This reaction here requires a polymerase to do the bulk of the work. Now, polymerases need a specific amount of salt, a temperature, a pH. There's an ideal condition to make them work properly. The same way that I need a proper temperature. I work fine at room temperature, but if you put me in Antarctica, I will freeze to death. The same kind of thing's going on with the polymerase. It wasn't at the right pH, so it wasn't working. So all that time wasted because I didn't know that unique bit of chemistry. Which brings me to a very important point, something that I run into with young biologists a lot, is, they think they just need to understand the biology and they'll be fine. It's not true, because you run into these sorts of problems all the time, and you need to have a solid understanding of the science as well as creativity to solve these problems. If you just want to be a pretty good scientist, you got to know chemistry. If you want to do really cool stuff, you need to know physics too. Now, this reaction here that I'm talking about, it's very slow and arduous. And what our lab is trying to do is take that and use engineering and photophysics to make that reaction happen very quickly, so we can test thousands upon thousands of chemicals to find one that inhibits this type of cancer activity. So what we're trying to do is use physics to make this much faster. So you got to know physics too. So if you want to do interesting things, learn enough chemistry. If you want to do amazing things, you got to learn physics too. So learn as much as you can and do awesome science. And don't make this mistake. Learn about tris. You got to know it all. So interesting stuff. And my incubation over there is almost done, so I get to leave for the day. And I'll see you guys later. Bye.